Hello friends, ACE Botany next episode. In this episode, we'll be discussing under NCERT quick reference, molecular base of inheritance 7th part, which mainly describing the replication 2nd part, original procedure. And 3 items will be the introduction, time, steps. And all references based on reprint edition NCERT March 2024, December 2024. So December 2024 edition is recent. I think I declare for convenience, I will share the screen so it will be easy for you. We can save time also. I think a screen came, so we can proceed. Okay. So finally, it is established DNA replication is semi-conservative. And it's paid. First, we are discussing because some values we have to take care. DNA polymerase enzyme is highly efficient enzyme, catalyzing polymerization of large number of nucleotides in a very short time. That is why efficiency. E. coli has got 4.6 into 10 raised to 6 base pair. That is E. coli. In humans, you already know that is within bracket 2 and 6.6 into 10 raised to 9 base pair. So E. coli with the 4.6 into 10 raised to 6 base pair completes the process of replication within 18 minutes. That means average rate of polymerization, it has to be 2000 base pair per second. NCERT is the base. NCERT page number 90, repeat edition March 2024, December 2024, recent edition also the given. And remember, it may be under optimal conditions, 18 minutes. Actually, E. coli chromosome needs 38 to 40 minutes. Different kind of replication is there. So generally 38 to 40 minutes. E. coli cell divides in every 20 minutes. Cell divides in every 20 minutes in nutrient-rich medium. These are our NCERT linked values. Now we can proceed to basic steps. 5 steps will be there. Origin of replication. 1. Activation of deoxyribonucleotides. 2. Unwinding of DNA helix. 3. And formation of primer strands. Important. Primer strand and elongation of new strand. I think idea clear. Now first page is clear, I think, so we can proceed to second page. Origin of replication. Now the event is replication 4. So due to high energy need, ender DNA cannot be separated on its ender lengths. Common question. Equal DNA where the replication originates, it is called ori. In prokaryote, single ori normally. Single ori is the important one. And eukaryote it is many ori. In E. coli it is ori C. Ori act as initiation site for the replication of DNA. I think idea clear. This is a diagram from NCERT. Next page it will be more clear. So second step, activation of deoxyribonucleotide. Second step, why activation needed? So for converting deoxyribonucleotides from where? From their monophosphate form to their triphosphate form, which is very important for DNA replication. See, from monophosphate form to their triphosphate form, which is very important for DNA replication. Monophosphate I given here, DAMP, DGMP, etc. Triphosphate, ATP. Here I am, mono, here T, tri. For this, what all things needed? Energy needed, no doubt. Phosphate needed, no doubt. Phosphorylase enzyme also needed. Now, third step, unwinding of helix. For unwinding process, a famous enzyme called Helicase, sometimes often refers to be unwindase. For us, Helicases is involved, causing separation of 2 DNA strands. I think the page is clear, so we can proceed to third page. See, this enzyme needs ATP. Unwinding will produce a Y-shaped structure. See, here you can find a Y-shaped structure. Replication 4, naturally, single strands of DNA will be unstable. So here it becomes single strand initially. So it is stabilized by SSBP, single stranded binding protein. And the DNA separation needs huge amount of energy. So that is not practical. So that is why from a point it starts. I think clear. While unwinding, normally a tension will be created on strand. So it is nullified by normally topoisomerase in eukaryote, topoisomerase. And in prokaryote, it is DNA gyrase. Topoisomerase basically. Now this diagram is important one, labeling question, theory and runs. Now formation of primer strand. Primer means preparatory or starter or initial or stepping, etc., on which other things can be added. After unwinding of DNA, respectively, new DNA strands has to be synthesized. Okay. In E. coli, DNA polymerase 3, which is true replicase, cannot initiate DNA synthesis. This is a headache. Means it is not able to deposit first nucleotide in a daughter strand. For this, there is an enzyme called Primase. Synthesizes a short RNA primer strand. Okay. On this, DNA will be added. That is how errorless replication proceeds. Later means after the initiation of DNA synthesis is over, primer RNA, that is strand is removed by DNA polymerase 1, again important. It is always called convergent enzyme. RNA synthesis involved in replication, initiation or replication of DNA, RNA primer and RNA primase. Now, elongation of a new DNA strand. Here we can find. Here, DNA has to be added. Enzyme involved is DNA-dependent DNA polymerase. But it can polymerize only one direction, that is only in 5' to 3', that is the rule. Since two strands are there, complication will be there based on that, because one is on 5' to 3', other will be 3' to 5'. So how this issue solved? It's a real big headache in molecular biology. So finally, a Japanese scientist called Okazaki solved this issue by giving an idea, maintaining the direction for enzyme, suggesting the replication is. That is why this diagram clearly shows it. Replication is continuous, because direction perfectly tally with 5' to 3' on one template strand with the polarity 3' to 5', on which new strand will be 5' to 3', producing leading daughter strand. I think idea clear. Now, in other strand, replication is not continuous, which is otherwise called discontinuous, in the form of short fragments called Okazaki fragments on the second template strand with the polarity 5' to 3', called lagging strand. So this is newly synthesized. This is segments. And remember, next is a data still. In prokaryote, Okazaki fragments contain 1,000 to 2,000 nucleotides. Eukaryote, it is around 100 to 200 nucleotides. Just the data given, that's all, not in NCERT. Every time when Okazaki fragments are formed, each fragments need RNA primer, and enzyme also needed RNA primase for each strand. Later, Okazaki fragments are joined together by enzyme DNA ligase. That is why ligase. DNA replication is semi-conservative and semi-discontinuous, important one. Then DNA replication takes place in S phase. Again, important, cell cycle may be studied. So this procedure should be highly coordinated or can lead to abnormalities, including polyploidy or even mutation. I think idea clear. If you like this episode, subscribe, ACE button, like it, share it, comment on it. We'll be continuing this series with another topic soon. So take care, bye, thank you.